Hello students, welcome to Vedantu NEET English. This is Basuraj sir, your biology master teacher. I hope everyone is safe and sound and doing extremely well in life. Now, this is gonna be one short revision of all the botany diagrams from your class 11th, because we know we have chapters like Living World, which is very easy. Then we have Morphology, Anatomy, little complicated. Then we have Cell, the cell cycle. Then we have chapters like your Plant Physiology. So all of these chapters in one shot today we'll be doing revision. So quickly let me know in the chat right now if I'm audible and visible to each and every one of you. Quickly let me know in the chat, students. Quickly let me know in the chat. And hello each and every one of you. Welcome to the class. Pratibha, Disha, Dreamer, Anshul, Wakanda. Why is the Black Panther here? Okay, but okay. So all of you, welcome to the class. Now students, I want all the bot army, right, entire bot army to show the energy, because this is a class where you will take away a lot of marks, and I guarantee it. Especially from plant physiology, you will take away at least 50 marks from this video right today. 50 marks, and that is not a joke. 50 marks is not a joke, students. I want everyone to promise me that you will stick around till the very end, and you will take away those 50 marks, and you will never regret in your life right now. Okay, so with that being said, students, the first particular chapter is Living World. Generally you get one question or two questions. Okay, generally you get one or two questions from this chapter, and it's relatively easier. And in this chapter there is a single diagram. There's a single diagram from this chapter. If you can see right here, this is the single diagram given in this particular chapter. Now students, I will ask you all of you very simple question. Ready all of you? The most simple question. Tell me students, in which particular section or which particular category do you find more number of organisms? Is it kingdom or is it species? Where do you find more number of organisms? You will tell me, sir, obviously in the kingdom the number of species, the number of organisms is going to be higher. It's going to be higher. Here we will have less organisms. But will the question will come from there? No. The question will come from a particular point called as similarities. Which particular category has higher similarity or lower similarity? I'll give a quick example. You will tell me the answer. Okay. Now, for example, sponges and humans. All of us belong to a single kingdom. All of you agree with me? Our sponges and humans belong to a single kingdom called as your animal kingdom. Yes. I don't know if you're plants. If you're plant, then plant kingdom. If you're plant, then we'll send you to plant kingdom. Okay. Now students, tell me, do you think a sponge and a human, do we have similarities? Do we have high number of similarities or less similarities? Tell me in the chat quickly right now. You will tell me, sir, obviously humans and sponges, the similarity at kingdom level, the similarities are comparatively lesser here. It's less here. But at the same time, different species of mangoes, similarity is higher here. So similarities is going to be comparatively higher. Similarities is going to be comparatively higher at species level, while at the same time at the kingdom level similarities are going to be less here. Now if that point is clear to all of you, diagram is done. Diagram is done, students. Diagram is done. Okay. Now next thing is called as plant kingdom. Very simple diagram on the board. How many of you know the answer? Try shooting the answer and go in the chat. I want chat to be on the fire. I want every single diagram, whatever you know about this diagram, one word in the chat quickly. Whatever you know about this diagram, quickly one word in the chat. So for example, if you know this diagram right here, both the diagrams belong to a particular algae called as green algae, not red, not brown. Now within the green algae, this right here is an example of colonial algae, like apartments, right? All of you stay in an apartment. This is an example of volvox. This is an example of, students, volvox right here. Volvox. Not Volvo, volvox, colonial algae. Now this right here is a filamentous algae. How many of you know filamentous? Filamentous means thread-like structure. Filament. Okay, it's a filament-like structure. This here is an example of Eurothrix, not Spirogara. This is an example of eulothrix. Both of these are examples of green algae. Cleared, all of you? Clear. Now I'll show you one more diagram. Tell me, students, when you look at this diagram right here, what comes to your mind? First of all, tell me. The first thing which comes to your mind should be, so this right here is a brown algae. This right here is a brown algae, while these two examples are red algae. Okay, done. Now within the brown algae, if you notice right here, students, within the brown algae, if you notice right here, this is a typical structure of your brown algae. The first one is example of your laminaria, fucus and dictyota. Laminaria, fucus, dictyota. That is BLP. Right? That this is going to, sorry. This is laminaria, this one right here. This is fucus and dictyota. That is LFD. In LFD, students, if you notice, every single structure has three main parts. Apart from fucus, which has your air bubbles, the three main parts here is, this right here is a root-like structure called as holdfast. The holdfast helps in attachment to the substratum. Then we have stipe, which is a stem-like structure. This is called stipe. Then we have frond. The frond is like a leaf-like structure which helps in photosynthesis. So holdfast, stem, and leaf. Every single example of your brown algae, same thing is going to be there. Now next thing is called as the red algae. In red algae, the two examples given is Porphyra and Polysiphonia. PP is the example, very simple. Porphyra, Polysiphonia. Now here you can see much more branching can be seen here, but Porphyra, not much of branching. Clear? Now, next thing is an example of. Tell me, students, what comes to your mind when you look at the diagram right here on the board? This is a diagram of a pteridophyte or bryophyte. Tell me in the chat quickly. Tell me in the chat. This particular diagram is a bryophyte or is it a pteridophyte? You'll tell me, sir, it is a bryophyte, liverwort. Okay. And especially if you know a particular structure which has a gemme cup, all of you will tell me this right here is an example of Markantia. Students, Markantia thallus. This is an example of Markantia right here. Now, students, I'll tell you a very simple thing. All the doctors, right, and all the future doctors, listen to me very carefully. I'll tell you one very simple thing right now, yet it is confusing to many students. The labeling which is given here, that is the confusing part. Antheridophore, Archegoniophore, these are not the sex organs. I'll repeat again. Archegoniophore, Antheridophore, these are not the sex organs. Nope. Antheridophore and Archegoniophore are technically the stalk. What are they? They are the stalk. They are the stalk. For example, a leaf has a stalk petiole, a flower has a stalk pedicel. So this also is a stalk. Then what is the sex organ here? This Archegoniophore will have Archegonia or Archegonium here. Antheridophore will have Antheridia here. So Antheridia is a sex organ. This is the stalk here. Now this is the thallus of a Markantia. Now if you know, all of the students, if you know, a Markantia is a liverwort. The main thallus here, is it erect or prostate? Tell me in the chat quickly, all of you. Is it erect or prostate? If you see here, the entire thalli is prostate. It is flat. It is flat on the surface. It is not erect here. It is flat. Okay. Apart from that, can you see rhizoids here, which help in anchorage here? Rhizoids. We can see rhizoids here also. Apart from that, there is something called as gemme cup, which helps in asexual reproduction. How? Gemme cup will produce gemme bud. It is an asexual bud which helps in asexual reproduction. Okay. So apart from that, nothing is very difficult here. Everything is very easy. Okay. Now here many questions will come. I want everyone to focus extremely high here. For example, this is an example of bryophyte again. But these are the examples of mosses. The first example given is Funaria. Example is given as Funaria. And this is an example of sphagnum. Example of sphagnum. Now students, what comes to your mind when you think of sphagnum? Tell me in the chat quickly. Any ecological importance comes to your mind? Any ecological importance? Sphagnum, I feel something for packaging material, for bandages, for use in agriculture also, use in growing plants. Anyone? Any ecological importance of sphagnum? Peat moss. Peat moss, exactly. So this is a peat moss which is used it has many ecological benefits. Sphagnum. Now students, if you see the Funaria, the main plant body is divided into the gametophyte and sporophyte. Now, the most important part here, I want everyone to clearly understand here is the gametophyte is free living. Gametophyte is independent. How? Green color, right? Green color. So, gametophyte is independent. Sporophyte is dependent on the gametophyte. Sporophyte is on the top, is dependent on the gametophyte. And also, the gametophyte is haploid. The gametophyte is haploid while the sporophyte is diploid here. Diploid. This is the main plant body. It's the main plant body. Main plant body. So in the case of bryophytes, the gametophyte, which is haploid, is the main plant body, which has the rhizoids. On top of gametophyte, sporophyte is developing. Sporophyte is dependent on the gametophyte. Okay, sporophyte is dependent on the gametophyte. That is very important. I hope all of you understand this point. Understand that point. Sporophyte is dependent on the gametophyte. A sporophyte has three main parts. That is foot. Right here is the foot, seta, and capsule. These three are the main parts of the sporophyte. These three are the main points about sporophyte. Okay, so sporophyte is dependent on the gametophyte. Sporophyte is diploid. Gametophyte is haploid. Gametophyte is the main plant body. Now I feel all of you are able to understand very. Yes, Wakanda gametophyte is autotrophic in nature. Gametophyte is able to produce. It is independent. It is independent. Okay, main plant body is independent. It is green in color. It has chlorophyll. It has chlorophyll. Now sphagnum here. Now this is a diagram they'll give you. Now the question is very simple. This diagram right here of sphagnum. Option number A, sporophyte. Option number B, gametophyte. Option number C, both A and B. Option number D, none of the above. This diagram they'll give you and they'll ask you, this diagram right here is the sphagnum sporophyte, gametophyte, both A and B, or option number D, none of the above. Tell me what is the answer quickly in the chat, all of you. See, if you look at the NCERT legend, it says sphagnum gametophyte right here. Can you see? Sphagnum gametophyte. So this is the main plant body of the sphagnum, which is independent. It can survive on its own. It means this structure right here is haploid. It is independent. It is independent and it is haploid in nature. So such small detail can be missed out. Small details can be missed out from the diagrams, which they can exploit, teachers. Okay. Now students, tell me, what comes to your mind when you think of this diagram right here? This diagram right here. What is this diagram? Tell me, is this diagram right here, is it going to be a pteridophyte or bryophyte again? Tell me quickly in the chat, all of you. This diagram right here, is it a sporophyte or is it a gametophyte? That also they can ask you. They can ask. But first of all, tell me, this diagram right here, is it a pteridophyte or is it a bryophyte? Quickly. Now you will tell me, sir, this is a diagram of a typical diagram of a pteridophyte. Typical diagram of a pteridophyte right here. And this diagram right here, students, I'll give you a small clue. I'll give you a small clue. This particular plant shows heterospory. Now tell me the answer. This diagram right here shows heterospory. Now tell me, heterospory. Now when you think of heterospory, there are two names should come to your mind. And one of the names is on the board right here. Okay, exactly. This right here is a diagram of your Selaginella. Example of Selaginella. Can you see? Selaginella is a diagram here. This is a pteridophyte. It has root-like structures. leaf-like structures and stem-like structure. Okay. Now here, if you remember, in the case of pteridophytes, what are the leaves called as? The leaves in the pteridophytes are called as fronds. They are called as fronds. Now, some fronds can be photosynthetic in nature. Certain fronds can be photosynthetic in nature, and they can also help in reproduction. Some fronds can help in reproduction. Such fronds, which are special fronds, also called as fertile fronds, they are called as sporophylls. They are called as sporophylls. Sporophyll are the fertile fronds. They are the fertile fronds. Now, students, if you know what is a sporophyll, right here, can anyone tell me what exactly is a strobilus then? Strobilus. I'll give you a simple explanation. Understand. Now, this right here, for example, these are my notes. Forget that. But for example, this is a sporophyll. This is a sporophyll, and this is sporophyll. If multiple sporophylls, they will combine and they will make a structure like this. Can all of you see this? If multiple sporophylls, they join together, multiple fertile frond, they join together, they end up making a structure called as cone. And this particular cone has another name. The cone has another name that is called as strobilus. It is called as strobilus, also called as cone. That is, many sporophylls are forming a structure. Okay, I hope all of you are able to understand. Okay, this is a structure of ecosystem. C is fern, D is salvinia. Now, students, tell me, salvinia shows homospory or heterospory? Tell me in the chat quickly. Salvinia right here, does it show homospory or heterospory? Quickly in the chat, answer. Homospory or heterospory? Quickly. Correct. Same example was given here also. Salvinia shows homospory or heterospory. Exactly. It shows heterospory. Okay. So heterospory is shown by Salvinia and Selaginella. Okay. I hope all of you are able to follow right now. Now, the last diagrams given your NCERT for the following gymnosperms. This here is a structure of cycas. And how do I identify cycas? The main distinguishing feature between a cycas and a pinus is the stem. If you look at the stem of a cycas, it is completely unbranched and single solid. If you see here, I hope it is visible to all of you. This right here in the middle is the stem. It is not visible in the diagram, but in my Telegram channel, I have sent you an image of a cycas very recently. The stem is completely unbranched here. At the same time, if you look at the pinus tree, the stem is branched. The stem is completely branched here. It is branched. Okay. Leaves are also changing. Here we have needle-like leaf. The last example is Ginkgo biloba, which is also called as a living fossil. Okay. Living fossil. Clear, all of you? Now, with that, plant kingdom is also over. It's over. Okay. So I believe till now you do not have any doubt, and the class is going very smoothly. In a similar fashion, we will finish the entire one shot of all the diagrams from botany in today's class. Okay. So all of you ready for the next chapter? All of you ready for the next chapter? All of you ready? Ready. Morphology of flowering plants. Now again, students, in morphology, many times NEET examination, questions have come from diagram, especially for placentation, aestivation, and also from the perigynous, hypogynous, epigynous condition. Recently, this year, NEET 2024, you got a diagram based question from the chapter morphology. So morphology is the chapter where they can combine questions from the diagram and examples and make a baby and give you. Okay, so diagrams from here onwards are going to be extra important. Okay, now morphology first diagram on the board right here. Now, you might not get a lot of questions from here, but let me ask you a question here. Let me ask you a question. Now, students, I want everyone in the chat to play a small game. Ready, all of you? One small game. Small game. The game is very simple. You're supposed to identify which of the following are vegetative parts here. Go. Which of the following are vegetative parts? Vegetative parts. So you tell me, sir, root is a vegetative part. Okay. Root. Then stem is a vegetative part. Leaf is a vegetative part. Even the, that's all right. Root, stem, leaf. Those following are the vegetative parts. Yes, vegetative parts. Reproductive parts or product of reproduction. Fruit is a product of reproduction. Fruit is a product of reproduction. Flower is reproductive part. Yes, bud also can help him. The bud is vegetative. Bud is vegetative in nature mostly. Okay. Now, that's all. Not many questions. You will not get many questions from there, but you will get a lot of questions from here, right here on the board. Okay. Now, I have not included the modification, so you can all of you can relax there. Okay. Now, the first diagram on the board. Tell me, students, is this diagram right here developing from radicle or a plumule? Developing from radicle or plumule. So, if you know this is a root, and root develops from a radicle. All of you know that. A particular root develops from a radicle. Now, this here is a typical structure of a taproot. Typical structure of a taproot on the board. Taproot. Okay. Taproot on the board. Now, I want everyone to quickly smash that like button right now if you're enjoying this session. Okay. Taproot. Now, if you see the taproot, students, this particular taproot directly develops from the radicle. Directly from the radicle. Now, this is called as the primary root or the main root. Okay. From main root, we can see some secondary root or lateral roots arising. Lateral roots or secondary roots. This is one type of root. The second type of main root is called as adventitious roots. Adventitious roots. Now, what is adventitious roots? Adventitious roots is a roots which is developing from any other part other than the radicle. Understand? It is developing from any part other than the radicle. Other than radicle. For example, leaf. You might have seen, right? Bryophyllum. On the bryophyllum leaf, on the leaf there is roots. From the stem, roots can arise. They can arise, right? Now, what is the fibrous root then? Understand? Very simple. I'll show you a diagram. If this is the stem right here, and we have nodal regions here, nodal regions. This right here is called as base of stem. Base of stem. From the base of stem, roots will start to arise. From the base of the stem. Such roots which arise from the base of stem, they are called as fibrous root. They are called as what? Fibrous root. So can I tell? Technically, can I tell? Since fibrous roots are arising from any other part other than the radicle, so can I call them as adventitious roots then? Yes, sir. I can call them adventitious roots. So fibrous roots are also a type of adventitious roots. Done. Now, this is a typical structure of a root. It has 4 main parts, but if you remove the root cap, 3 main parts. Now, the most important part here is the root cap. Can you see here? This is a thimble-like structure. Called as root cap. Now, you might ask me, sir, what is the function of root cap? The main function of root cap is protection. This root cap right here, it completely protects the region of meristematic activity. Region of meristematic activity is completely protected by the root cap. Now again, what is this region of meristematic activity? Very simple. Region of meristematic activity is a region on the root which has small thin-walled cells which undergo rapid division. Repeat after me, all of you. Meristematic region of meristematic activity is a region which has small cells, actively dividing cells, thin-walled cells with dense protoplasm. Okay. Now, then is the region of elongation. In region of elongation, the cells will elongate. Cells will elongate. Okay. And region of elongation is also the region. It is also the region responsible for length of root. Length of root. Okay. The last region is called as region of maturation. The cells are maturing here now. Nice beard is coming. Can you see? When the cells mature, they get a beard. And the beard is called as what? Root hair. It's called as root hair. So as the person matures, they get beard. And here we have the region of maturation, where the cells mature and they have a very fixed function and structure. And on the epidermis, there is formation of root hair. Formation of root hair. Okay. So I hope all of you are following till now. How is the revision coming along? Very super revision, no? This is called as super revision. Now, do you realize when I'm speaking something, I'm actually quoting the lines of NCERT. How many of you realize that? Raise your hands. How many of you realize? Raise your hands. When I'm telling a statement, I'm telling the statements of directly lines of NCERT. Now, this is what I want from all the students also. I want all of you to connect and get the reference which I'm saying. Because when you read the lines of NCERT at least around 5 to 6 times minimum, you will reach the stage of following every single line of NCERT. Okay. So I hope all of you are able to follow which I'm saying. Because when I'm saying the lines, I'm quoting NCERT lines. Quoting NCERT lines for all of you. Okay. Now, next diagram is called as a leaf diagram. Now here I'll ask you questions. Ready? I'll ask you questions here. Now there are 3 main parts here actually. 3 main parts. The first part is called as the leaf base. Leaf base. Second part is called as the petiole, and the third main part is called as lamina or leaf blade. Lamina or leaf blade. Can you see? This entire thing called as lamina or leaf blade. Now question is very simple. Tell me which region of the leaf is responsible for photosynthesis at a higher level. Photosynthesis higher level, which one? Tell me. You will tell me, sir, lamina is the answer. Lamina is the broad part which is responsible for photosynthesis. Okay. Now, apart from that, what is petiole? Petiole is a stalk-like structure. Stalk. You know, it's a stalk. Now, one more question I can ask you. Okay. In the case of dicot, reticular venation, in the case of dicot, petiole is made up of dash. In the case of dicot, petiole is made up of dash. Dicot. What is petiole made up of? Very simple question. Is it parenchyma, collenchyma, sclerenchyma? Now, students, remember, a petiole is very flexible. All of you run tomorrow morning, go find one leaf, try to bend the petiole. The petiole is very flexible, elastic in nature. It is able to bend and again come back to the shape. It also flutters in the wind. That is petiole. And the petiole is made up of collenchyma. Because collenchyma can provide flexibility, rigidity at the same time, and also photosynthetic in nature. Okay. Then we have the axillary bud here. Understand, all of you, maximum effort, ultimate focus. Just 2 minutes. See this. If this is a stem right here, stem. A particular stem has the nodal regions. Yes, it has the nodal regions. At the nodal regions, what develops? We have a leaf at the nodal region. We have a leaf here. At a nodal region, we have the leaf. Okay. Now, this particular region right here, can all of you see this? This is called as leaf axis. It's called as leaf axis. Okay, leaf axis. Now, within the leaf axis, when the shoot apical meristem is growing, some cells are left behind. When the shoot SAM, shoot apical meristem is growing, some cells are left behind, and those cells are called as bud here. And this is the bud right here, present in the leaf axis. This bud right here is called as axillary bud because it is present in leaf axis. Axillary bud. Now this axillary bud right here can develop into a branch or a flower. That is the axillary bud right here. Okay. Now, what is stipule? Stipule is a leaf-like structure, folding of young leaf, which helps in protection. Now, the next thing is leaf base. Very important. Leaf base can be swollen. Leaf base can be swollen, and such a leaf base is found in leguminous plants. Can anyone tell me what is the swollen leaf base called as? Swollen leaf base is called as pulvinous leaf base. It is called as pulvinous leaf base. Clear? Now, one more thing I have to tell you here is, if the leaf base can partially or completely cover the stem, then what is it called as? I'll give you a small hint. It is present in the case of monocots. Where the leaf base partially or completely covers the stem. Such a leaf base is called as sheathing leaf base. It is called as what? Sheathing leaf base. Students, do you realize I am teaching you every single point of NCERT in this rapid revision of mine? Diagrams. I am not just telling you the diagram, but I am teaching you the concepts here and there. Okay. Now. Very simple diagram, very simple, very easy. Venation is basically arrangement of veins and veinlets. Here we have two types. The first one is called as B, that is reticulate venation, parallel venation. Reticulate venation is found in dash plants. Reticulate venation is it found in the case of monocot or dicot? Monodicot. Quickly tell me the answer in the chat. Quickly answer in the chat. Reticulate venation is found in the case of monocot or dicot. In the case of dicot. Parallel venation in the case of monocot. Now here I'll tell you very important thing, phyllotaxy. Understand phyllotaxy. I will teach you phyllotaxy and you will never forget in your life. Okay, ready? Now if this is the stem right here. Here we have the nodal regions. Nodes. These are the nodes. At the nodal region, we can find only one leaf. Alternatingly, if you find only one leaf, such phyllotaxy is called as alternate phyllotaxy. It is called as alternate phyllotaxy. But, what is the but here? The but here is, if at the nodal part, if you find two leaf, such an arrangement is called as opposite phyllotaxy. So only one, alternate, two, opposite. Okay. Now what if I add more than two at a single node? Here also. More than two at a single node. Such phyllotaxy becomes world phyllotaxy. It becomes world phyllotaxy. A single leaf is going to be alternate. Two leaf, opposite. Many, world. Now here they have given you certain examples, but if you are my student, if you are part of bot army, why botany? Okay, very simple. Not much thinking is capacity there, but if you are part of bot army, you can tell me easily the other examples. For here, China rose, they have given you one example for alternate. Now, can anyone give me other examples of alternate phyllotaxy? Anyone? Alternate phyllotaxy, other examples in alternate quickly. China rose, one is from M, one is from S. China rose, mustard and sunflower. China rose, sunflower and mustard. Come on, both army, let's come on. Next one. Opposite, guava and calotropis. Very simple. World, only single example, that is Allostonia. Allostonia is a simple example of world phyllotaxy. Okay. So I believe all the bot army is able to answer all this one. Okay. Now next is simple examples of racemose inflorescence and cymose inflorescence. Now I'll give you a simple trick here. Okay. Now all of you know what is running race. All of you know running race, and all of you play running race also, no? All of you have tried running race in your school. All of you have tried running race 100 meters. Okay. Now I'll give you a scenario. Okay, I'll give you a small scenario. There is a running race between a particular one boy who is around, let's say, 16 years or 18 years of age, one boy, or a man who is around 20 years of age or 25 years of age. One more male is there who is around 60 years. One is 20 years, one is 60 years. Tell me, who will win the race? Who will win the race? Will 20 years old man or 60 years old man? Who will win the race? You will tell me, sir, the young one will be able to finish the race faster because he's younger, more energy, more muscular development. At that stage, he'll win the race. Same concept apply here. In racemose race, racemose is the race. Racemose inflorescence. In this entire race, who will win? Young one will win. So young ones at the top, older flowers at the bottom. All of you understood? Young flowers at the top, older flowers at the bottom. Such an arrangement is called as acropetal succession. Acropetal arrangement. It is called as what? Acropetal arrangement. Acropetal arrangement. So older flowers at the top, younger flowers at the top, older flowers at the bottom. Older flowers at the bottom. Now this will continue, keep on growing. It will keep on growing. There is no terminal. There is no terminal flower. At the same time, cymose inflorescence. Cymose inflorescence, understand. Cymose inflorescence terminates with a flower. It terminates with a flower. Can you see? The flower. Old flowers at the top. Old flowers at the top. Basically reverse. Young flowers at the bottom. Young flowers at the bottom. Such an arrangement is called as basipetal arrangement. It is called as what? Basipetal arrangement. Basipetal succession, basipetal arrangement. Okay. Terminates with the flower. Very simple. All of you should remember this. Racemose and cymose. All of you will remember. Yes or no? Tell me quickly in the chat. All of you will remember this? Yes, you can. Okay. Now, next thing is going to be the hypogynous, perigynous, and epigynous. Now, students, I will teach you with the help of diagram first. Just focus for the next 1 minute. I'll teach you the entire thing. 2 minutes. Let's say 2 minutes. I'll teach you the entire thing. Ultimate focus. Because in NCERT this year, NEET 2024, there was a question where they give you these two options and you were stuck. You were stuck. Okay. So listen to me very carefully now, all of you. Listen to me very carefully. This diagram right here. This is the gynoecium. This is the gynoecium right here. Gynoecium. Gynoecium is on the top. Stamen, petal, calyx, all the different whorls are coming from the bottom. Can you see? Every single whorl is arising from the bottom, and the ovary is superior here. Ovary is superior, gynoecium on the top, like a queen on the top, on the throne here, on the throne. Such a condition is called as hypogynous condition. Hypogynous condition, represented by capital G. G on the throne. Now, if you see the perigynous condition, platypus peri, okay, perigynous condition, the gynoecium here is in the center. Gynoecium is in the center right here, center, and all the different whorls are arising from the rim of the thalamus. This is the thalamus. This is called as a rim of thalamus. It's called as rim of thalamus. So petal, sepal, stamen. Every single other whorl is arising from the rim of thalamus. Gynoecium is in the center. Such a condition is called as perigynous condition. Perigynous condition is going to be what? Half inferior. Last one is called as epigynous condition. In epigynous condition, ovary is gone, completely engulfed. For example, all of you, if this is the gynoecium, let's say this is the gynoecium, entire other whorls are completely covering. They are completely covering the gynoecium. This is the gynoecium. Let's say bottle as gynoecium. This is the gynoecium. All the other whorls are completely covering it. All the whorls are completely covering the gynoecium. Can you see here, all of you? If you notice here, this is the gynoecium in the middle. All the other whorls, entire thalamus also, thalamus is completely covering the gynoecium here. and all the whorls are on the top, and the ovary is inferior ovary here. Inferior ovary. Now, quickly examples. Ravindrani is already telling me the answers. Okay. Ravindra is telling me all the answers already. Now, examples for hypogynous. Quickly go in the chat. Examples for hypogynous. Hero must have brinjal. Hero, hypogynous, must, mustard, have, hibiscus, brinjal. So China rose, hibiscus, same thing. Brinjal and mustard. 3 different examples. Now what about perigynous condition? People pluck pink roses. People, perigynous, pluck plum, peaches, roses. So roses, peaches, plum. Last one is epigynous, epic. Epigynous, epic. Sunny goes camping. Epic, Sunny goes camping here. Tell me the answer quickly. Guava goes camping, cucumber, and ray flowers of sunflower. So epic, Sunny goes camping. Epic, Sunny, sunflower goes camping. Guava and cucumber. Yeah, guava and cucumber. That is the examples of perigynous inferior ovary. Inferior ovary. Done? Done. Now, students, I will ask you a very difficult question now. Very difficult question. Okay. Tell me which is the outermost whorl in a flower. Which is the outermost whorl in a flower? Quickly, go. Outermost whorl in a flower. The outermost whorl in a flower. Let's see who can tell me. Outermost whorl is calyx. Calyx. What is calyx made up of? Calyx is made up of petals. Second is corolla. Corolla is made up of petals. Andrecium. Andrecium is a group of stamen. Gynoecium is a group of pistil. Pistil. Okay. So, outermost whorl calyx, corolla, andrecium, gynoecium. Those are the 4 different whorls of the flower. Now, students, how many of you want to learn sexual reproduction of flowering plants with me? Never. Students, I've never taught you, right? That is one of the chapters in botany which I have never taught you, but let me teach you that also. Okay. So very soon I'll be teaching you sexual reproduction of flowering plants as a botanist to all of you. Very soon, don't worry. And also even molecular basis. That also is coming up. Don't worry. Okay, it's coming up. Now, next thing. Now this is called as aestivation. This is called as aestivation right here. Now what is aestivation? Very simple. Aestivation is very simple. Okay, understand all of you. Aestivation is nothing but arrangement of petals, arrangement of sepals. I'll repeat again. Aestivation is nothing but arrangement of petals or arrangement of sepals with respect to each other. For example, the first one is called as valvate aestivation. In the case of valvate aestivation, all the petals and sepals are arranged on a margin to margin. They are just touching each other, just touching each other. Can you see? But they are not overlapping. They are not overlapping here. No overlapping. Next one is called as twisted aestivation. In twisted aestivation, there is a constant overlap. There is a constant overlap. Can you see? One petal or sepal is overlapping on the other one, and they're alternating manner, in an alternating manner. That is called as twisted aestivation. Next is called as imbricate aestivation. Imbricate. In the case of imbricate aestivation, there is overlap. There is overlap, but the overlap is not in a particular order. So if you can see here, this sepal is completely on the inside, while this petal or sepal is completely on the outside. So completely on the inside, completely on the outside. Remaining 1, 2, 3. Remaining 3 are in an alternating fashion. In, out, inside, outside. So one completely on the inside, one completely on the outside, and remaining are alternating. Okay. Remaining are alternating. Last one is called as Wechsler aestivation. Now to all the bot army. One question to all of you. What is the other name of Wechsler aestivation? Let's see who can tell me. What is the other name of Wechsler aestivation? That is called as papilionaceous corolla. It's also called as papilionaceous condition. Okay, papilionaceous. It is mainly found in the case of Leguminosae family. Okay, now, or Fabaceae, which what do you call it? Now here, if you can see, this is called as standard wing and keel. Standard wing and keel. This is entire thing is 1 plus 2 wing plus 2 keel. This right here is a Wechsler estivation. Now examples, again examples are very important. Now valved estivation, calotropis, valved chair. Twisted estivation, a lady from China has a twisted cotton. A lady from China has a twisted cotton. So lady, lady's finger, China, China rose, twisted, twisted estivation, cotton, cotton. Okay, lady from China has a twisted cotton. Imbricate, advocate, advocate fights gun cases, Gulmohar and Cassia. Wechsler estivation, PP vaccine, B and peen, B, bean and P. Okay. Next is all the different types of placentation. What is placentation? Placentation is arrangement of ovules. Arrangement of ovules inside the ovary. Inside the ovary. Okay. On the placenta. Arrangement of ovules inside the ovary on the placenta is called as placentation. Placentation. First is called as marginal pea plant. This is called as the ventral suture. On the ventral suture, there is arrangement of ovules in a marginal way. Then the second one is called as axial placentation. Here the entire ovary is divided into multiple segments. You can see there are multiple chambers here, and all the ovules are present on the center, arranged on the placenta. The same goes for parietal, but parietal peripheral. So parietal means peripheral. Peripheral, all the ovules are arranged on the periphery. Can you see here? All the ovules are arranged on the periphery here. Now this right here, this diagram is parietal placentation. It is usually unilocular, but later in the plant's life, it becomes bilocular. Why? Because in later stage in the plant's life cycle, there is a formation of false septum called as rhapulum. False septum called as rhapulum formation happens. It becomes bilocular eventually. Next is called as free central. All there is no septa formation here. Same thing with no septa. Okay. Last is called as basal placentation. A single ovule is present at the base, called as basal placentation. Again, examples are going to be a best friend here. Let me tell you again and again. So botany, tell me. Botany, tell me what is the example of marginal placentation example quickly in the chat. Marginal placentation examples quickly, all of you. Marginal, pea plant. Okay. Axial, allow China to leave. Allow axial placentation. 2, tomato to leave. Right? Lemon. Next. Parietal placentation. Parents are masters. Argemone and mustard. Free central. Fredo, change your DP. Dianthus and primrose. Basal placentation. Birds migrate south. Marigold, sunflower. So if you can remember all of these examples, no one can stop you from scoring really well in this chapter. No one can stop you. Literally no one. So remember the examples. That is the key here. Okay. And I have made a tricks for it. All of you know the matrix. Now this is a typical example of a fruit. Can anyone tell me which type of fruit is it? Is it a pome? Is it a pepo? Is it a berry? Is it a atrio of berries? Or is it a drupe? In the chat quickly. Which type of fruit is this? Which type of fruit is the following? All of you quickly tell me in the chat. Exactly. This right here is a drupe. Now the entire pericarp is divided into 3 main layers. Epicarp, mesocarp and endocarp. Epicarp is the outermost layer. Mesocarp is the middle layer. Now mesocarp can be 2 types. Mesocarp can be fleshy in the case of mango, or it can be hairy in the case of coconut. Mesocarp is hairy here. Okay. This entire thing is the mesocarp. Mesocarp. Inner carp is called as endocarp. Endocarp is stony. Stony. Here, look up this stony. Then there is seed. Then there is seed. It is developing from a monocarpally superior ovary. It is developing from a monocarpally superior ovary. They can ask you a question there. Next is a seed. Very simple seed. This is a typical structure of a dicot seed. You can see here there are two cotyledons. One, two. Two different cotyledons. There is plumule. Plumule will. This entire thing is called as what? This entire thing is called as embryonal axis. Embryonal axis. In this entire embryonal axis, there is something called as radicle which develops into the root. Plumule develops into shoot. And entire thing has a seed coat. Seed coat has two main parts called as testa and tegmen. Testa on the outside, tegmen on the inside. Then there is a scar here called as hilum. Hilum is a point of attachment to the fruit. Then there is a small pore here. Small pore through which water can enter inside called as micropyle. Called as micropyle. Through which water can enter inside. Micropyle. Students, do you realize I am quoting line by line of NCERT. I am quoting line by line of NCERT and I hope you are able to follow. Any doubt, you can ask me. Okay, shoot it out in the chat. No one should be shy. Okay, now hilum is a scar. Yes, hilum is a scar. Okay. Next thing is the monocot seed. Now here, which type of question we can expect here? Let me tell you, the main question which they can ask you is the single cotyledon. What is the name of a single cotyledon present in the case of monocot? Can anyone tell me? Which is the single cotyledon present here? Single cotyledon, single cotyledon, single cotyledon. We will do genetics one more time, don't worry. Something is coming up for all of you students. Something is coming up for you, for all the YouTube students. Something big is coming and it's going to be revealing very soon. Okay. Which is the cotyledon here, students? Single cotyledon. Single cotyledon here. In the case of monocot. Can you see here, students? This right here is the single cotyledon present here. Okay. Shield shaped structure. Single cotyledon. Entire thing is covered by seed coat and fruit wall. Now, why have they written seed coat and fruit wall? Because both are fused here. Both are fused here. Okay. Now, entire endosperm is separated from the cotyledon with the help of a protein layer called as aileron layer. Protein layer called as aileron layer. Endosperm right here. Endosperm, which helps in developing the embryo. Nutrition. Now, within the embryonal axis, this is the embryonal axis. There is radicle and plumule. Now, radicle and plumule has bodyguards here. Radicle and plumule has bodyguards. Who are the bodyguard here? Not Salman Khan, but you have other bodyguards here. Understand? Coleoptile, P and P. Coleoptile plumule. Coleorhiza are radicle. So, coleorhiza is a bodyguard for radicle. At the same time, plumule has a bodyguard called as coleoptile. Okay, coleoptile. Clear? Beautiful. Easy. And with that, morphology of flowering plants is over. Students, do you realize this is called as rapid revision? And let me tell you, students, again one more time. If you attend this rapid revision, you could have solved right now. You could have solved so many questions from morphology. I bet you could have solved many questions from morphology if you understand the rapid revision. Okay. Now, for anatomy, students, for anatomy, I'll teach you not the entire thing, because if I teach the entire diagram for anatomy, 1 hour will go off. Okay. So, I'll tell you the main points here. Rest, I'll tell you where to find. Okay. Now, can anyone tell? It's given here only. Okay. Students, don't see this. All of you don't see this. Okay. Tell me which is monocot, which is dicot here. This right here is a diagram of the stomata. Which is for monocot, which is for dicot. Don't see. No cheating. Okay. Which is for monocot, dicot? This is A. This is B. Tell me which one is for monocot. Monocot. stomata, monocot stomata. This one right here. Can you see? This is the stomata of monocot B. This is a dumbbell. All of you go to gym, you can tell me this is a dumbbell right here, right? Dumbbell. This right here is a dumbbell. Don't know, dumbbell. Okay. Now, if you see here, this entire stomata is dumbbell shaped here. Dumbbell shaped. At the same time, here in the case of dicot, it is bean or kidney shape. Bean or kidney shape. Bean or kidney shape. Okay. Now this entire stomata has multiple parts. For example, these structures right here, which opens and closes, these structures right here are called as guard cells. Guard cells. Guard cells have the chloroplast. They have chloroplast. Around the guard cells, there are certain cells called as subsidiary cells. Subsidiary cells. And these cells, outer cells, are called as epidermal cells. Epidermal cells. So, and the middle part is called as stomatal pore. Stomatal pore plus guard cells plus subsidiary cells is part of entire apparatus here. Okay, guard cells. So every single time there is a water entering inside, every single time water is entering inside, guard cells will swell up. They're going to swell up. It will open. When all the water is going away, guard cells will become flaccid. They'll become flaccid. Water in, turgid. Water out, flaccid. That is called as opening and closing of stomata. Opening and closing of stomata. Now, this is the different types of arrangement in the case of the vascular bundles. In the case of vascular bundles. Okay. Now, if you see here in this particular diagram, first one, all the vascular bundles are present in its own radii. See, one radii, one more radii, one more radii. Xylem and phloem are present in an alternating manner, and the same radii, such as found in the case of root, it is found in the case of root, it is called as your radial arrangement. Called as radial arrangement. Radial arrangement. The second one, can you see? Xylem and phloem are present on same radii. Xylem present like a sandwich. Xylem, phloem present together, holding hands. Okay. Such an arrangement is called as your conjoint arrangement. Conjoint. Conjoint. Now, if there is cambium, someone is third wheeling here. Okay. Between xylem and phloem, there is a cambium in the middle. Such a arrangement is called as open. If cambium is present, it is called as open. If cambium is absent, it is called as closed. Closed. Closed is mainly seen in the case of leaf. This is mainly seen in the case of open, is seen in the case of stem. Stem. Why? Because cambium is responsible for the secondary growth in a plant. Secondary growth in a plant. Okay. Now, students, for entire, let me tell you again, entire diagrams of monocot versus dicot. Monocot versus dicot, it has been covered in 1 hour 45 minutes in detailed in this video right here. So go to the channel and type monocot versus dicot. You will get a complete revision of monocot stem, dicot stem, monocot root, dicot root, monocot leaf, dicot leaf. The entire diagrams is decoded in this particular video, so go find it today only. Okay. Now, but apart from that, I'll ask you simple questions. Okay. Tell me A or B. First of all, tell me this particular diagram, is it for root or is it for stem or leaf? Tell me. Is this diagram for root, stem or leaf? Simple question for all of you. This diagram right here, is it for stem, root or leaf? You will tell me, sir, I can see root hair here. I can see root hair here. So this is root. This is root right here. Now, second, next question. Tell me which of the following, A or B, which of the following is for monocot and which one is a dicot here? Quickly in the chat. Which is monocot, which is dicot? Is A monocot or dicot? Is B monocot or dicot? Quickly in the chat. I'll give you exactly 10 seconds. Tell me which of the following is monocot, which is dicot. The clue, I'll tell you a small clue. The clue lies in the xylem and phloem bundles. Here only 4. Here we can see polyar condition. We can see polyar condition here. A is going to be dicot. A is going to be dicot. So B is going to be monocot. It is going to be monocot. Okay. Why? Because of the vascular bundles. If you can see here, there is tetrarch condition here. Can all of you see? There's a tetrarch condition here, but here we can see polyarch condition. Polyarch condition. Okay, polyarch condition here. Polyarch condition, that is more than 6 xylem bundles. Okay. Now tell me, students, this particular section, is it for a root? Obviously not. Is it a stem or leaf? Stem or leaf? Quickly go. Stem or leaf? And students, quickly smash our like button already. All of you, smash our like button. 111 votes, but only 70 likes. What is this? Quickly, let's make 100 likes. This is a section of a stem. Tell me, students, which one is monocot, which is dicot here? Is A monocot or B monocot? Or which is monocot, which is dicot? Quickly go. What is A, what is B? I'll give you a small clue. Look at the vascular bundles arrangement. Look at the vascular bundles arrangement in the case of monocot and dicot. You can tell me the answer. Which is monocot, which is dicot? First one is dicot. Lock. Can we lock? Yes, this is dicot. This is monocot. Why this is monocot? Because of the scattered vascular bundles. And can you see here? Water cavity is present here. Water cavity is present here. This is a dicot because of the ring arrangement. Because of the ring arrangement of vascular bundles. Okay. Apart from that, you can also see the cambium here. Cambium. The cambium is absent in the case of monocot. Absent in the case of monocot. Entire cortex divided into hypodermis, cortical layer, and endodermis here. Endodermis. No such differentiation. No such differentiation here. Now tell me, this is a leaf section. Which one is monocot, which is dicot here? Which is monocot, which is dicot in the leaf arrangement? Which is monocot, which is dicot? Very easy, you can tell me. Very easy. Okay, I'll tell you, I'll increase the difficulty. Tell me which one is dorsiventral here, A or B? Which is dorsiventral? Is A dorsiventral or B dorsiventral? Very simple to all of you. Tell me which one is dorsiventral here, dorsiventral, A or B? Which one is dorsiventral? What is the meaning of dorsiventral? The meaning of dorsiventral is that dorsy, 2, ventral. So ventral side and dorsal side. In ventral side and dorsal side, there is two different types of parenchyma here. Okay. Here if you see, there is palisade mesophyll here, and here if you look at the bottom, we have spongy mesophyll. So two different types of parenchyma, that is mesophyll. Two different types of mesophyll here. Dorsiventral. They're also called as dicot. But in the case of monocot, it is isobilateral. Why do we call it isobilateral? We call it isobilateral because same types of mesophyll entire. Same type of mesophyll. There is no differentiation. You will not find spongy, palisade, nothing. Single type of mesophyll present here. Single type of mesophyll present here. Okay. You can also find air cavities here. Stomata. Okay. Stomata can also vary. For example, in the case of dicot, more stomata at the bottom. More stomata at the bottom. But in the case of monocot, the number of stomata is equal on both the sides. It is equal on both the sides. Okay. So with that, anatomy also briefly it is over. Okay. The next topic is called a cell chapter. Now students, after cell, I'll give you a small break before we start plant physiology. Okay? All of you agree with me? Now I want ultimate focus again. Ultimate focus, because cell the unit of life is very easy, very simple diagrams, very easy diagrams to go. Can we start? Yes, all of you? Can we start now? Very simple it is, very simple. Cell the unit of life. Can we start, all of you? Cell the unit of life. Now after this, I'll give you a small break so you can go relax also. But right now, let's finish cell cycle and cell unit. This entire unit will finish. Before plant physiology, I want everyone to fresh your brains up and come back. Okay? Structural organization, I will take someday, but not today. Not today, but I will take it someday. Okay? Gopika ma'am has taken it, right? Now, this is a typical diagram of different types of cells. Okay? First, we have the red blood cells, white blood cells, different types of shapes. Can you see here? Here we have columnar epithelial cells, mesophyll cells. Did we see that right now? Here, mesophyll, same mesophyll cells. Same mesophyll cells are also present here. Then we have tracheids here. Can anyone tell me tracheids are part of xylem of phloem? Tracheids are part of xylem of phloem. Anyone? Tracheids, xylem, nerve cell, longest cell. Longest cell. A question to all of you. A question to all of you. Actually, a difficult question. Okay, a very easy question. Tell me, in which cell of the human body only glycolysis takes place? Answer is on the board. Answer is on the board. Which cell in the human body where only glycolysis takes place? There is no Krebs cycle, there is no link reaction, nothing is there. No ETC. Which is that cell? Which is that cell where only and only glycolysis takes place? Answer is on the board. Go. It is gonna be the red blood cells. Red blood cells are completely biconcave and they do not have nucleus. Nucleus is missing. Mitochondria is missing. If mitochondria is missing, where will the Krebs cycle takes place? Does not happen. So there is no Krebs cycle. There is no electron transport chain in the case of red blood cells. Only and only glycolysis takes place. Why? Because glycolysis occurs in the cytoplasm. Okay. Next is the shape of the cells. All of you can see. Here we have typical eukaryotic cell 10 to 20 micrometer. Typical bacteria 1 to 2 micrometer. PPLO 0.1. Viruses 0.02 to 0.2. Now I'll ask you a very simple question. For example, let's take one volunteer from the chat. Any volunteer? Let's take Ami. Let's take Ami. Okay. Let's take Ami for example. Okay, Ami. Now Ami is standing here. Okay. Now here I'm adding a filter. I'm adding a filter here. The size, the pore size of the filter. The pore size of the filter, let's say, is around 1 micrometer. 1 micrometer. Okay, pore size. Now can the virus enter? Can the virus enter, students? 1 micrometer filter is there. Can the Ami get damaged? Can she get the virus? Can the virus pass through? Yes, sir. Because virus size is 0.02 to 0.2. Can the PPLO enter? Yes, PPLO also can enter. PPLO can also enter. So remember that. For 1 micrometer grid, they can enter easily. So viruses cannot stop. Viruses need a very proper filtration unit. Okay. Now, question to all of you. This right here, the diagram on the board, is this a plant cell or an animal cell? Go. Is this a plant cell or an animal cell? Quickly answer in the chat. Is this right here a plant cell or is it an animal cell? Yes, all of you. Quickly. Just look at it. Let's look at it. A small clue, let's say, look at the outer layer. Look at the outer layer with the answer. One of the biggest clues right here. One of the biggest clues, the right in the middle also. This one is one of the clues. This is one of the clues. Exactly. This right here is a typical plant cell. This right here, is a typical plant cell. Why? Because the first of all, cell wall. Second of all, chloroplast. Third is going to be large central vacuole. Large central vacuole. Three different points here. And this is a typical animal cell. Why? Because there is something called as centrioles. These centrioles are missing in plant cell. They are missing in plant cell. Now, how many of you know how to ride a bike or a car? Raise your hands. Raise your hands quickly. How many of you know how to ride a bike, or do you know how to ride a car? Quickly, tell me. Quickly, all of you. How many of you know how to ride a bike or a car? Or a bicycle also works. Bicycle also works. Okay. Now, for example, student, for example, you're riding on the bike. Easily you're going. Okay. Suddenly one hump will come. One pothole. Road is in Indian roads. Simple Indian road, when you go, what are the chance of finding a pothole? Extremely high. The probability of finding a hump, extremely high. Probability of finding a good road, less. Okay. So when you're riding, hump down. So that is a typical Indian road, at least near my house. Okay. You know who took inspiration from there? Do you know where is it inspired from? Right here. This is the Indian roads are inspired from a typical plasma membrane. Typical plasma membrane, also called as cell membrane. Why, sir? Why the inspiration? Start driving a bike here. One hump, randomly humps are there. Such a condition is called as mosaic condition. It is called as mosaic condition, inspired from our Indian roads. Okay, it is completely random. You can see randomly. You can see the proteins. We can see the proteins, sugar, cholesterol, fat, lipid. Randomly they are arranged here. Okay, randomly they can be integrated. Okay, and this is a typical structure of a plasma membrane. Now, if you see the plasma membrane, students, here we can see the lipid bilayer. This entire thing is called as the lipid bilayer here. Entire thing is called as lipid bilayer. Lipid bilayer. Polar head and non-polar tail. Polar head and non-polar tail are arranged here. This is called as lipid bilayer. Entire thing is called as lipid bilayer. Okay. Okay, it's called as lipid bilayer. Entire thing. Now you can see here proteins also. Proteins are there. Now proteins are mainly found based on two categories. Okay, based on ease of removal. I'll give you a simple example. This right here is a, let's say this right here is a stupid paper also, not rotating only. See here. Now, for example, this is a protein. This protein is somewhere outside like this. Now, can I remove the protein easily? Excalibur. I can remove the protein easily. I can Excalibur. Chosen one. Now, what if the protein is completely on the inside now? Protein is completely inside. Tell me, is it easy to remove or difficult to remove now? It is going to be very difficult to remove now. Somewhere have to put inside and difficult, you have to pull it out. Difficult to remove. Based on the ease of removal, we have the integral protein, very difficult to remove. It is completely on the inside. Peripheral protein, present on the outside, very easy to remove. Okay, so peripheral proteins present on the outside, very easy to remove, while the integral proteins are on the inside, very difficult to remove. Done. Such a good example. Okay, phosphobilayer is the entire lipid. Okay, now this is a typical example of ER, also called as endoplasmic reticulum. Endoplasmic reticulum mainly divides the entire cell into luminal, extraluminal, that all of you know. Okay, extraluminal, luminal, extra, all of you know. But here diagram doesn't say that. Diagram shows you some of the ER which has endoplasmic reticulum plus ribosomes. Such a ER is called as RER, rough endoplasmic reticulum. Second type, there is an ER plus no ribosomes. Plus no ribosomes. Such a ER is called as SER. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Okay. Smooth endoplasmic reticulum. SER. Rough ER produces proteins. Smooth ER produces lipids and also certain hormones. Lipids and hormones. Okay. Now this is a typical example of Golgi, but very difficult to understand. I will show you a better example. Okay. I will show you. Why is the cable blinking a little? No? Blinking. Now see here. This is the structure of a typical Golgi body. Listen to me very, very carefully now. Very carefully. Listen to me. Here, if you see this diagram of a Golgi body, it has two different faces. Like some of you have, right? In front of your friends, you have certain people in your life now. In front of you, they'll say something. Behind your back, they'll say something. They will do that. Okay. Yes, HDMI 2, the cable is little not working. Okay. Now see here, students. This particular Golgi body has two faces. The first is called as cis phase, also called as forming phase. Other phase is called as trans phase, also called as maturing phase. Cis phase also called as forming phase. Trans phase is called as maturing phase. Now, any single protein or lipid formed by the ER, listen to me very carefully. This particular ER, rough ER or smooth ER, they're producing proteins and lipids. These proteins and lipids are taken up by the cis phase. So all the proteins and lipids are entering through the cis phase. Now what is happening here? Once the proteins and lipids enter through the cis phase, they undergo packaging. Okay. So all of them starts getting packaged, and all the proteins and lipids are removed outside. So if the protein is entering, it'll become glycoprotein out after this. If a lipid is entering, it'll become a glycolipid after Golgi. So what is Golgi? Golgi is like Amazon factory. It'll help in packaging. So cis phase is the entry, trans phase is the exit. Okay. Cis phase, protein is entering, glycoprotein is coming out. Lipid is entering, glycolipid is coming out. All of you can agree with that. Okay. So this is cis phase and trans phase, cis and trans. And this foldings, can you see foldings are called as cisternae. Okay. Cisternae. Now this is an example of a typical double membrane structure called as mitochondria. Lord mitochondria. Now this mitochondria is also called as, can anyone tell me what is the other name of mitochondria? Anyone knows? Anyone? It is also called as powerhouse of the cell. Mitochondria. Why? Because in the case of mitochondria, with the help of oxidative decarboxylation, right, and also other reactions, there is production of oxidative phosphorylation. Because of ATC, there is ATP produced here. ATP. Oxidative phosphorylation, ATP is being produced here. Now, since a double membrane, there is outer membrane, inner membrane. There is also in this mitochondria, there is also DNA, and also there is protein formation, and also there is a ribosomes here. This foldings of the inner membrane is called as crista, which helps in increasing the surface area. Okay. Entire thing is called as matrix intermembrane spaces. There is also a matrix here. Next, this is a chloroplast. Chloroplast is also a double membrane structure. Outer membrane, inner membrane, all of you can see. Now here, each individual structure is called as a coin-like structure is called as thylakoid. The entire stack is called as granum. This granum and this granum, Both are connected to each other, like how they're holding hands. For example, this and this are holding hands. The hand is called as stroma lamella. Stromal, lamella, stroma, lamella is the connecting link. Is the connecting link. Entire thing has a matrix called as stroma. Stroma is the matrix. And within the stroma, dark reaction, thalocode and grana, stroma lamellae. Here we have light reaction. Light reaction. Light reaction, dark reaction. Okay, dark reaction. This is a typical example of ribosomes. There are two types of ribosomes, 70S and 80S. Very simple, understand. Very simple. First of all, let's understand eukaryotic one, 80S. 80S is divided into a larger subunit and a smaller subunit. Larger subunit in 80S is called as 60. It is called as 60S, 60S and 40S. In the case of prokaryotes or in the case of prokaryotes or in the case of mitochondria chloroplast, 70S ribosome. 70S ribosome has a smaller unit, larger unit. Larger unit is 50S and 30S is a smaller subunit. 30S is the smaller subunit. Now what is S here? S is called as Sedgwick unit, also called as sedimentation coefficient. Also called as sedimentation coefficient. Okay. Now this is a structure of this is an electron microscope, normal structure of cilia and flagella. First of all, understand students, what is cilia and flagella made up of, and where are they present, and how are they? What is the use of them? Cilia and flagella helps in motility. Okay. Now cilia and flagella, students, understand very carefully. Cilia and flagella, entire structure is made up of a cytoskeleton. I'll repeat again. The entire structure of cilia and flagella is made up of a cytoskeleton called as microtubules. Made up of microtubules. Very important, understand, very important. Listen to me very carefully. Listen to me, students, all of you, listen to me. I'll show you a small video here. Okay. Oh, I forgot. I have to download the video. I didn't download it, students. So next time I'll show you. Now this is the entire structure of a flagellocilia. This entire structure is called as axoneme, also called as core. Okay, axoneme or core. This entire axoneme or core has outer layer called as plasma membrane. Okay. Now here there is arrangement of microtubules. On the periphery there are totally 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 periphery microtubules. Peripheral microtubules are 9. In the center there are 2 microtubules. Okay. Central microtubules, peripheral microtubules. Now the peripheral microtubules are attached to center with the help of radial spoke. Attachment point is called as radial spoke. This is called as the bridge, and this red part, can you see? That's called as central sheath. It's called as central sheath. Not given here, it's given here. Central sheath. So arrangement here is what? The arrangement here is 9 plus 2 arrangement. Centriole is 9 plus 0 arrangement. Okay. Connecting between the two different types of microtubules is the interdoublet bridge. The entire peripheral microtubules are present in doublets. Can you see? 1, 2 doublets, AB, doublets. Now this is typical structure of a chromosome. Now students, in chromosome, why have they marked kinetochore here? What is the kinetochore? Kinetochore is a structure. On the present around the primary constriction. Also called a centromere. Around the centromere. What is the importance? For example, this is the primary constriction. This is the P arm and Q arm. Chromatids. Around the centromere, there is a primary layer, proteinaceous layer. This is called as kinetochore. The kinetochore. Now, what is the function here? Kinetochore is the point of attachment of all the spindle fibers. So all the spindle fibers all the spindle fibers attached to the kinetochore, not to the centromere. This is centromere. Okay, centromere is the middle one. These are different types of chromosomes. This is P arm and Q arm. P arm and Q arm both are equal in metacentric, submetacentric, telocentric, acrocentric, and telocentric. See here. Metacentric both are equal, V shape. V shape. VLJI. VLJI. Submetacentric, also called as L shape. J shape is acrocentric. I shape is telocentric. VLJI. VLJI. So with that, the cell unit of life is also done for the revision point of view. Now I'll be teaching you something called as cell unit of life. So sorry, cell cycle, cell division. Here I will not teach you the entire mitosis and meiosis because that will take 2 hours. For example, if I teach you the entire cell cycle, which I have completed the entire one shot lecture of cell cycle and cell division in just 2 hours 8 minutes with questions, with PYQs, with questions. This is done cell cycle and cell division in just 2 hours. So please watch this lecture, because if I show you the diagram of cell cycle, entire thing I have to teach you. Okay, but I'll teach you something. I'll teach you what is this structure here. What is this? Focus. Small thing it is. Okay, very small thing. Okay, now understand this part. Okay, understand this part here. Entire cell cycle is broadly classified into 2 categories. Entire cell cycle is classified into 2 broad categories. Okay, cell cycle or cell division, cell cycle, not cell division. First is called as interphase. Other one is called as M phase. Interphase is further divided into G1, S, G2. Gap 1, synthesis, gap 2. M phase is divided into 2 other categories. Karyokinesis and cytokinesis. Karyokinesis is a division of nucleus. Cytokinesis is division of cytoplasm. Karyokinesis is further divided into PMAT. What is PMAT? PMAT is nothing but prophase, metaphase, anaphase, and telophase. Cytokinesis again there are 2 types. In animal cell it is different, where we find furrow formation. In the plant cell also it is different, where we find what? In the plant cell we find cell plate formation. Now if you see the diagram, students, this entire cell cycle is divided into 2 parts. This entire yellow part, this entire thing is part of interphase. Can you see interphase? So 95% of time a cell is technically at interphase. It is at interphase. Now remaining part, can you see the black part? This is called as the M phase. This entire dark blue part, can you see a purple or violet color? This entire thing is karyokinesis. Prophase, metaphase, anaphase, telophase. Pink color is called as cytokinesis. So this entire thing is cell cycle, interphase, G1S, G2, interphase, M phase, karyokinesis, small strip called as cytokinesis. So that is the entire cell cycle. Okay, entire cell cycle. Now students, the last unit, 3 main chapters. Diagram revision today. Okay, so I believe entire pace is going really well. Now time is, let's take 8:40. Let's take a break until 8:40, 8:55. Let's take a break till 8:50. Okay, let's take a break till 8:55. Okay, 15 minutes break and then we will start. And then we'll start the plant physiology. p.m. Okay. After that, we will be starting. After that, we will be starting the entire thing. After that, we'll be starting the plant physiology. So all of you quickly smash that like button. Smash that like button. Until then. Okay. So I'll see you exactly at 8:55. Until then, take care. I'll come back and we will do the plant physiology after that. Okay. So see you around, all of you, quickly. Come back at 8:55. Sound check. So, hello students, welcome back to the amazing class of diagrams. Now, we finished almost all the different chapters of your class 11th. We started with living world, plant kingdom, morphology, anatomy, cell unit of life, cell cycle and cell division. Now, we'll be starting off with something called as plant physiology, all the three chapters in one shot, starting right now. Okay. Now, students, quickly let me know in the chat right now if I'm audible and visible to all of you. Quickly let me know in the chat if I'm audible and visible to every single one of you. Quickly let me know in the chat, all of you. Quickly let me know. NEET English. Yes. Quickly let me know in the chat, all of you, if I'm audible and visible to all of you. Okay. Can we start, all of you? And all of you quickly smash that like button. Yes. Keep smashing that like button because it's very important, because it helps in the algorithm. It helps in YouTube algorithm. If you do not know why I asked you to like the video. Okay. Now let's start with the first chapter, that is photosynthesis in higher plants. All the diagrams, because there are many diagrams from this chapter. Many diagrams are there. Starting with the first diagram. Voice check, yes. Pass the voice proper, right? Okay. All the bot army assemble. Now, the first diagram on the board. Now, this diagram is from a famous experiment, which with the help of this experiment, we were able to identify oxygen or discover oxygen. And this was an experiment done by Joseph Priestley, called as Bell Jar experiment. Very simple experiment. Let me tell you. Okay, very simple experiment. In the first setup, in setup A, Joseph Priestley took a mice and a candle, and after some time, the candle and the mice, both of them died completely. In the setup B, he also kept a mint plant. He also kept a separate plant inside. But he noticed that even after some time, the candle is burning as well as the mouse also alive. What is the magic here? The magic is very simple. When the burning of candle or respiration by the mouse, it consumes oxygen, and the oxygen is being replaced by the plant here. That was the logic behind it. Again, very simple diagram, very simple diagram of the chloroplast given here. Chloroplast, if you can see here, it is a double membrane structure, outer membrane, inner membrane. Then there is a coin like structure. Can you see? Coin like structure is called as thylakoid. The entire stack is called as grana. They are connected to each other with the help of stromal lamellae. Then there is ribosomes here. Now, can anyone tell me, which type of ribosomes do you find in the case of chloroplast? Is it 70S or is it 80S ribosomes? Quickly in the chat, all of you. See, this is a chloroplast. It is a type of semi-autonomous structure. The ribosomes present here is 70S ribosomes. Unlike mitochondria, which is 80S. 70S here, just like prokaryotes. Then there is oil droplets, starch grains. The entire matrix here is called as stroma. Now, within the stroma, dark reaction takes place. Within the grana, the light reaction takes place. So, light and dark reactions takes place here. Okay. Now, this is a diagram. Also, typically it's a chart, but I've included here. It's a chart for your absorption spectrum, action spectrum. Now, what is the difference here? Your absorption spectrum tells you that which pigment is getting absorbed at which wavelength. Which pigment, chlorophyll B, chlorophyll A, carotenoids, is getting absorbed at which wavelength. Okay. Now, what is action spectrum? Action spectrum tells you that at which wavelength photosynthesis is happening. At which wavelength photosynthesis is occurring or happening. That is action spectrum. Absorption spectrum, at which wavelength absorption is happening. That is absorption spectrum. Now, students, if you notice here, there are two main peaks here. This is for chlorophyll A. One peak is here, one peak is here. Now these two main peaks are the regions of red light and blue light. These two are the main regions of, let's say this is blue light and this is red light. Whenever a plant is given red light and blue light, whenever a plant is given red light and blue light, rate of photosynthesis is much more higher. How do you find it? If the photosynthesis is higher, more oxygen. That's all. Find at which point, find at which wavelength of light more oxygen is being released. It means that in that particular wavelength, photosynthesis is happening at much more faster rate. Okay. Now, if you overlap the absorption and action for chlorophyll A, wherever there is more absorption, there is photosynthesis. Wherever there is less absorption, less photosynthesis. More absorption, more photosynthesis. So this tells you that whenever a pigment is getting absorbed at blue and red, blue and red is getting absorbed more. In blue and red, there is more rate of photosynthesis. That is the overlapping. That is the overlapping between the action spectrum and absorption spectrum on the board. Okay. Now this is a typical structure of a light harvesting complex. Now what is a light harvesting complex? First of all, it is a complex. All of you know what is a complex. You might have seen buildings, large buildings called as complexes. Within a complex, it has multiple levels. Similarly, here also within a complex, it has two main components. A complex has two main components here, two main components. The first one is called as a reaction center. It's called as reaction center. Other thing is called as antenna molecules. Antenna molecules. Now, what is antenna molecule? Now, I'll tell you a small scenario, very small scenario. Okay. There's one large area is there. Let's take my area only. In my area, I have one big dawn is there. One large gunda or a dawn is there. The dawn is called as the reaction center. Is the dawn reaction center. Okay, ultimate dawn. Our ultimate dawn has multiple right hand, left leg, right leg. Lot of underlings under him, small gundas are under him. He has. So reaction center is the main dawn. The antenna molecules are the right hand, left hand are the small dawn. Let's call them small dawn. Now, what is the importance here? Understand. Large dawn is taking over the entire area. All the information from the small dawn is going to the large dawn. Same thing is happening in the case of the light harvesting complex. In the light harvesting complex, all the energy is being absorbed by all the photons being absorbed by the pigment molecules or the antenna, and entire thing is transported to the reaction center, because pigment molecules or the antenna, they cannot do the reaction. They do not have the enzymes present for it, while the reaction center can do the enzymatic work and electrons can be excited. Clear, all of you? Clear? So this is the main gunda, this one, who has the ultimate power. Small gundas are giving the money to him. Okay, money to him. Clear, all of you? Clear? Very simple to understand, no? Very simple. My area name, don't even ask. My area, one ultimate dawns are there. Okay, so that's why I don't even go out. Now, here all the energy is transferred. Can you see? All the energy is trapped and sent to the main boss, and energy is going up. Energy is going up. Okay. Reaction center. Example, I'll tell you. Example. Reaction center example is chlorophyll A. Chlorophyll B, carotenoids, xanthophyll are the examples of antenna molecules. So this is the chlorophyll A. Chlorophyll A is the reaction center. Now here is a typical example of non-cyclic photophosphorylation, but I'll give you a better diagram from my side. Okay, see here. This is my diagram which I've shown you many times, and this is the most important and easy way to understand. Okay, I will explain the entire thing. Ultimate focus for the next 10 minutes. Ultimate focus time. Okay. Now, who is this chlorophyll A? Chlorophyll A is the big dawn, big gunda here, chlorophyll A. Small, antenna molecules. So all the antenna molecules are giving the energy to chlorophyll A. Okay, sunlight is coming, energy is being trapped, energy is sent to the chlorophyll A. Chlorophyll A, electrons are getting excited, and there is something called as primary electron acceptor. Example is pheophytin. Pheophytin is picking up the electrons. Okay. Now from pheophytin, electrons are sent to PQ, CY, PC. PQ, CY, PC. Repeat after me. All of you, repeat after me. PQ, CY, PC. Repeat again. PQ, CY, PC. What is PQ? Plastoquinone. What is CY? Cytochrome B6F complex. What is PC? Priyanka Chopra. No. Plastocyanin. Plastocyanin. So PQ, CY, PC. Electrons are being sent. One ATP is also being formed here. Then from PC, electrons sent to the second complex, that is PS2. Here is PS1. So here we have PS2 working at 680 nanometer. Here we have PS1 working at 700 nanometer. Two different photosystems. Systems are basically structures which are made up of light harvesting complexes. PS1. So PS2 to PS1. 680 to 700. 680 to 700. Okay. Now from here again electrons are being sent up to primary electron acceptor. That is the primary electron acceptor here is FeS, iron sulfur centers. Then FD, ferredoxin. FD is ferredoxin. Now finally from ferredoxin, the final electron acceptor. The final electron acceptor. Final electron acceptor from in the non-cyclic photophosphorylation is NAD+. NAD+ is the final electron acceptor. So electrons are sent here. Electrons are sent here. Now when electrons are sent here, this NAD+ gets reduced to NADPH2. NADPH2. In this entire reduction process, two protons are sent from the stroma to here. Stroma to here. So two electrons, two protons, two NADPH converted to NAD+ to NADPH2. Enzyme is FNR. Ferredoxin reductase enzyme. Ferredoxin reductase enzyme. Now I hope all of you understood big time now. I'll leave the board one more time. Take a screenshot, write this down and understand. Because in this entire reaction, students, electrons have to be constantly supplied. Electrons have to be constantly supplied. There has to be a source now. What is the source? Source is called as photolysis of water. In the lumen part, water is being broken down. Water is split. That is called as hydrolysis of water. When the water is split, hydrogen, oxygen, electrons, H2O, right? This hydrogen is basically sent here. Hydrogen is sent to the ATP formation. I'll teach you later on. It is sent in ATP formation. This is sent to the ATP synthesis. Oxygen is released as a byproduct. Electrons are sent here. So there should be a constant supply of electrons is coming from water. So water is splitting up. There is a constant supply of electrons. Okay, constant supply of electrons. Very easy, no? Tell me, students, very easy, no? Very easy. See here. Light harvesting complex, photosystem 2, 1. Electron acceptor, transport system, PQ, PY, PC. Complex 2, finally to NAD+. Finally to NAD+, NADPH. NADPH. Here, photolysis of water. Photolysis of water. Okay, photolysis of water is also happening. Okay, crazy it is. Crazy. Okay. Next. Once non-cyclic is over, next thing is called as cyclic. Even more simple. Cyclic is even more simple. Very simple. Again, chlorophyll A is the main boss. All the energy is being sent here. Yes. Electrons are excited. Primary electron acceptor, FeS, FD, PQ, CY, PC. Same thing again. But here, electrons are again sent back to the chlorophyll A itself. There is a complete cycling of electrons here. Cycling of electrons is happening here. Cycling of electrons is happening. Okay. Cycling of electrons is happening here. So only ATP is being produced here. But here if you notice in the non-cyclic, there is O2 is evolved right here. ATP is produced, NADPH is produced here. Can you see? NADPH is produced. Non-cyclic, Z shape, also called as Z scheme. It is also called as Z scheme. Z scheme. Okay. Now, very simple, very simple. Now, students, see, that was easy part. That was very easy. Okay. Now, the next concept which I'm going to teach you, it requires little bit extra attention, because chemiosmotic hypothesis. My dog also is a little barking. Okay. Chemiosmotic hypothesis is very easy once you understand it. Let me make it clear. It may sound difficult. It may sound difficult at first. Chemiosmotic hypothesis. But it is actually very easy to understand. Okay. Can we start? Can we start, students? Can we start? Can we start, students? Let me drink some water. Can we start? All of you drink some water. Water break. All of you drink some water quickly. Can we start now? Yes. Chemiosmotic hypothesis. Now, yes, Cash is little not caching today. Okay. Chemiosmotic hypothesis. Chemiosmotic hypothesis explains how ATP is being produced. Chemiosmotic hypothesis explains how ATP is being produced. For example, if you see here, I told you ATP is being produced here. I told you here also ATP is being produced here. ATP is produced. But how? How is the ATP being produced? That concept is explained in how the ATP is being produced is explained in the chemiosmotic hypothesis. Okay. Now, what is the structure? Next thing is, what is the structure right here? Understand. All of you remember the chloroplast? In chloroplast, there are thylakoids. Entire stack is called as grana. Entire stack is called as grana. Yes. There were multiple thylakoids. This ring right, can you see this ring? This ring right here is one such thylakoid. If you take one thylakoid and expand it, if you see it under microscope, it is called as this structure. This structure. Okay. Now, if you see here, PS2, PS1, PQ, CY, PC. Do you remember, students? Do you remember this? Do you remember somewhere? PQ, CY, PC. Do you remember it, students? Here. PQ, CY, PC. Plastoquinone cytochrome B6 subcomplex, plastocyanin. PQ, plastoquinone. CY, cytochrome B6 subcomplex. PC, plastocyanin. So this right here is nothing but non-cyclic photophosphorylation. Non-cyclic photophosphorylation is occurring right here on the lumen, on the thylakoid. Okay. Now, for this entire process to occur, there are four main requirements. For this entire process to work. The first one is called as a proton pump. Second thing is intact membrane. Second thing is called as intact membrane. And the third is called as ATP synthase. And the fourth and the most important part is nothing but the proton gradient. Now, these three, the first three are very easily available. Proton pump is there, it is there. Intact membrane, it is there. ATP synthase, it is here. So the first three criteria is easily fulfilled, very easily fulfilled. The last criteria, right, the proton gradient, this criteria takes a little bit of time, and this criteria is actually fulfilled by three different methods. Now, I'll recap what we learned right now. Listen to me very carefully. Chemiosmotic hypothesis is a process which explains, it is hypothesis, right? It explains how the ATP is being formed. Clear? Okay. What is the structure right here? The structure right here is the thylakoid structure. Thylakoid. Non-cyclic photophosphorylation is happening. Clear? Okay, there are 4 main requirements: proton pump, intact membrane, ATP synthase, proton gradient. First 3 are already available. Last one needs to be formed. Last one needs to be formed. Okay. Now, what is the meaning of gradient? Meaning of gradient is a difference. So we have to create now a difference in proton. For example, if you see the diagram right here, we can see here low proton in the stroma and high proton in the lumen. So there's a difference has to be created. High proton inside the lumen, low proton inside out in the stroma. Now, how do we create this difference? How do we create? There are 3 main ways that difference can be created. 3 main ways. The first one, understand. First one. Remember this: oxidation of water, also called as photolysis of water. H plus was byproduct. All of you remember? Yes or no? Tell me quickly in the chat. All of you. Here. Without here, right? Photolysis of water. Hydrogen, oxygen, electrons. Electrons were sent here. Oxygen was removed as a byproduct, and hydrogen was sent to ATP synthesis. This is the point right here. Can you see? This is the hydrogen sent here. So can I tell inside the lumen right now, the H plus concentration is increasing? It is increasing. Done. Now the second one. This structure right here. Where is it? Plastoquinone. This structure right here, plastoquinone. This plastoquinone is not only an electron carrier. It is not only an electron carrier, it is actually a hydrogen carrier. This particular plastoquinone can pull one proton from the stroma and pull it inside the lumen. Can you see? It's being pulled here. So can I tell, because of this process, the H plus concentration is increasing on the inside, and outside H plus concentration is decreasing because of this, because the H plus is entering inside now in the lumen. That is the second method, second thing. The third one, okay. Sir, how can we see all these reactions in the, because of there are certain tests are there to find out. The tests are there. Third most important one. Okay. In the final step, did I tell you NAD plus is getting reduced to NADPH? Reduction is happening. Now in this reduction, I told you one proton is being used here. So can I tell, because the usage, H plus concentration is again decreasing because of that usage in the stroma. The stroma, protons is decreasing, lumen, proton is increasing, and we have the proton gradient. Now once this proton gradient is created, after what happens? After that, this is the work of ATP synthase. Now all the H plus will pass through the ATP synthesis. Every single time 4 H plus is entering, 1 ATP is being formed. And that's how ATP formation, takes place in the plants in the case of the non-cyclic and cyclic photophosphorylation. That's how ATPs are being formed there. Beautiful, right? Beautiful. This is called as chemiosmotic hypothesis. Next is called as C3 cycle. All of you know the C3 cycle, famous C3 cycle. The famous C3 cycle. I will give you a small recap here, students. Understand how the protons are being made here. The first way is photolysis of water increasing. Second method, plastoquinone, hydrogen. Done. If it is pulling from here, it is decreasing on top, decreasing on the stroma. Third method, NADP is getting reduced to NADPH. Again, protons are decreasing. That is how the proton gradient is formed. Okay, C3 cycle. Now, what is C3 cycle, students? C3 cycle is a method in which we make organic molecules. See, plants has to make food. In light reaction, we make ATP, NADPH and everything. But in the dark reaction, plants actually make the food. So all the food which we eat is the main reason is C3 and C4 cycles, because this is the process in which glucose is being made, starch is being made here. Okay, Now understand. Here I'll teach you one small method. All of you listen to me. Listen to me. Small method is there called as 3, 2, 1, 0 method. Ready? Ready, all of you? Something called as 3, 2, 1, 0 method. 3, 2, 1, 0 method. Okay. Now what is 3 here? 3 stands for 3 main steps. 3 steps. And 3 ATP is being used here. 3 ATP is being used. What is 2? 2 stands for 2 NADH is being used. 2 NADH is being used. Now what is A1? The 1 stands for all of this is happening for 1 CO2. 1 CO2 is happening. 3 ATP, 2 NADH. Now what is 0? 0 stands for no energy produced. No energy produced. 3, 2, 1 method for you to remember it. 3, 2, 1 method. Now understand, all of you. Now what are 3 steps here? The 3 main steps is carboxylation, reduction, regeneration. 3 main steps. In carboxylation, CO2 is picked up by RUBP. Rubulose 1,5 bisphosphate plus CO2 happens here, plus CO2. And it undergoes carboxylation. To produce PGA. What is PGA? 3-phosphoglyceric acid. So RUBP plus CO2 will give PGA. First step is called as carboxylation. Now there is an enzyme involved here. The enzyme involved here is Rubisco enzyme. Okay. Carboxylase oxygenase enzyme is involved. First step. Now PGA gets converted. PGA undergoes reduction to produce triose phosphate. So PGA to triose phosphate. In this entire step, 2 ATP is used, 2 NADH is used. 2 ATP and 2 NADH is used in the reduction phase. Okay, 2 ATP and NADH. Now triose phosphate eventually gets converted to sucrose and starch, which can be used by us to eat and everything. Last step is called as regeneration, where triose phosphate is again converted back to RUBP. In this entire step, 1 ATP is being used. 1 ATP. So in total, 3 steps, 3 ATP, 2 NADH for 1 CO2 and 0 energy is being used in this entire process. That is 3, 2, 1 method. 3 to 1 method of the C3 cycle, also called as Calvin cycle, also called as PCR cycle, also called as dark reaction. Many names, one cycle, one reaction. Many names, one reaction. I hope all of you are able to follow. Plant physiology doesn't get easier than this. I will tell you confidently, if you learn the plant physiology from me, you will never go wrong. Okay. Now students, tell me, if this is for 1 CO2, how many ATPs? 1 CO2, 3 ATP and 2 NADH. Okay. For 1 CO2, 3 ATP, 2 NADH. How many for 1 glucose? How many ATP for 1 glucose now? How many ATP and how many NADH for 1 glucose? Simple example I'll tell you. For 1 CO2, 3 ATP. Into 6 you do. Into 6 again. Why into 6? Because for 6 carbon, 18 ATP, 12 NADH. 18 ATP, 12 NADH. Done. So into 6 you should do. Into 6. 18, 12 NADH. So very simple. 18 ATP, 12 NADH. Because this entire thing is for 1 CO2. But for glucose, 6 times we should turn it. 6 times. Next is C4 cycle. C4 cycle is also called as Hatch and Slack pathway. Okay, Hatch and Slack pathway, because of the scientists who were involved here. It is also called as your dark. It is also part of a dark reaction. Also called as C4 cycle. Okay, students. Now here, why is it called a C3 cycle? Why is this called as C3 cycle? Because the first stable product is 3 carbon. That is why C3 cycle. Okay, that is why C3 cycle. But here, this is happening in C4 plants. whereas there is no photorespiration. There is no photorespiration because the plants in your C4 cycle show something called as Kranz anatomy, where all the vascular bundles are completely arranged in a wreath-like structure. Okay. Now here we have two different types of tissues, two different types of cells actually, cells. One is called as mesophyll cell on the outside, on the inner side bundle sheath cell, inner side bundle sheath cell. Now outer mesophyll cell has a separate reaction, while bundle sheath cell also has a different type of reaction occurring here. Understand, all of you, understand. In the mesophyll cell, phosphoenol pyruvate, also called as PEP, phosphoenol pyruvate, also called as PEP, is the primary CO2 acceptor. So CO2 is entering, it is forming a bond with PEP. And the enzyme involved is pepcase enzyme, called as pep carboxylase enzyme. Okay, pepcase enzyme. Here we have pepcase enzyme. Pepcase. Now PEP plus CO2. PEP plus CO2, 3 carbon plus 1 carbon. It ends up giving 4 carbon compound that is called as C4 acid. C4 acid, that is OAA. Initial is OAA is the first product. What is OAA? Oxaloacetic acid. So CO2 plus PEP will give you OAA. Oxaloacetic acid. Okay. It gets converted to an acid called as malic acid. Malic acid transported. Can you see transported to bundle sheet. Malic acid. Now malic acid undergoes decarboxylation. It means malic acid will release a CO2. Okay. Malic acid will get converted to a C3 acid. Can you see? C4 acid, C3 acid. What is the difference? 1 carbon difference. 1 carbon is released out. Now this 1 carbon is entering the Calvin cycle here. Calvin cycle. So if I tell you, Calvin cycle occurs in all plants, true or false? True. Calvin cycle will occur in C3 plants. It will also occur in C4 plants. Can you see here? So Calvin cycle occur here. RUBP, triose phosphate, carboxylation, reduction, regeneration, everything will happen in the bundle sheet. The enzyme, that is Rubisco enzyme, is also present here. Beautiful, right? Beautiful. So C4, C3, one CO2 is released. That CO2 is taken up by Calvin cycle. So there is Calvin cycle. So Calvin cycle is occurring where? Calvin cycle is occurring in the bundle sheet of C4. Okay. Transport again back to C3. C3 again is regeneration to pepcase. Entire thing is C4. So with that, all the diagrams from the chapter photosynthesis in higher plants is covered now. Now we go to the next chapter, that is called as respiration in plants. And let me tell you, entire thing will be finished today. Okay. Entire thing will be finished today. Okay. Now next thing is called as respiration in plants. Can we start, all of you? Can we start, students? Can we start? 2 more chapters to go. Can we start? Oops. Can we start, students? Can we start now? Respiration plants. Tell me, how many of you find difficulty? You know glycolysis, but you are not able to solve the questions on glycolysis. Does it happen to you? That you know glycolysis? Glucose, glucose 6-phosphate, fructose 6-phosphate. You know glycolysis, but you are not able to solve the questions. Because this is a problem with many students. Because you have sometimes do not have an idea which type of questions you can get from glycolysis. Which type of questions? Listen to me today. Okay, listen to me. I will give you all possible questions from glycolysis. Can you see which type of questions can come from glycolysis? For example, in this entire glycolysis, they can ask you which is the rate determining step. All of you can hear me, students, right? All of you can hear me? Yes. Which is the rate determining step? Third step here. First step, second step, third step. Rate determining step. So I'll write down for all of you once again. I'll write down the steps for you. Okay. All the steps on the board. All the steps on the board. Third step in which fructose 6-phosphate is getting converted to fructose 1,6 bisphosphate. In the third step is irreversible. It's a redetermining step, which is irreversible step. Irreversible step is the first step, third step, and the 10 steps are the irreversible one. In which step water is being removed? Tell me, students. Which step water is being removed? In the ninth step, water is being removed. So 2-PGA to PEP, phosphoryl pyruvate. Ninth step it is being removed. Which step NADH is formed? Sixth step NADH is formed. Here to here, here to here, NADPH is formed. Which step NADH is being formed? Sixth step. They can ask you which step ATP is produced. ATP is produced. ATP is produced in the 7th step here and 10th step here. ATP is being produced, 7th and 10th. Or they can ask you in which step ATP is being consumed. First step, third step, ATP is being consumed. First step and third step. Which is the first enzyme? The first enzyme in this reaction is hexokinase. Last enzyme, that is pyruvate kinase. Last enzyme. This entire glycolysis is also called as EMP pathway. Why? It is called as EMP pathway because of the three scientists, Emder, Mayorov, and Parnas scientist. Which step oxidation happens? Oxidation happens in the 6th step here. NADH formation, 6th step. Does it happen in aerobic or anaerobic condition? It happens in both. It happens in aerobic condition and also anaerobic conditions. Crazy, no? So if you master all of these questions, there is a 99% chance that you will tell the right answer. Okay. So they can ask you questions which step, but if you should also know when I say third step, you should tell me fructose 6-phosphate to fructose 1,6 bisphosphate. That also you should know. Which is the first step, third step, fifth step. What is happening in the 9th step? In the 9th step, 2-PGA, 2-PG, phosphoglycerate to PEP, phosphoryl pyruvate. That also should be known. Okay, that also should be known. If you know this much right, glycolysis is over. You should never worry about glycolysis. Okay. Now that is the entire. Why so much of love in the chat? What do you want, all of you? What do you want? Tell me. Sudden suspicious love in the chat. Suddenly all the different color hearts. What do you want? Tell me. What do you want, all of you? Quickly. Why the muska? Why the buttering? Tell me. Why the buttering now? Okay. Now next thing I'll teach you something called as the anaerobic respiration. Anaerobic respiration. All of you remember this particular thing happens in both aerobic and anaerobic both. It happens in aerobic and anaerobic both the condition, that is glycolysis. First step in respiration. This structure, that is lactic acid fermentation, alcoholic fermentation, lactic acid fermentation and alcoholic fermentation, both of this reaction will occur in anaerobic condition. It will occur in anaerobic condition. Very simple to understand. Okay, very simple to understand. Glycolysis, glycerol phosphate, pyruvate, pyruvic acid. Entire thing is glycolysis. After glycolysis, some of them it can undergo lactic acid formation in lactic acid bacteria, muscle cells, or in the presence of yeast, brewer's yeast, it can get converted to ethanol and CO2. Now, which type of question they can ask you here? The main type of question which they can ask you here is the enzymes involved. For example, if you see here, CO2 is being released out here. So the first enzyme which comes to your mind is decarboxylase enzyme. It should be a alcoholic. It should be pyruvate decarboxylase. alcohol formation. Alcohol dehydrogenase. Two enzymes here are, first one is pyruvic acid decarboxylase enzyme. Second enzyme should be alcohol formation. So alcoholic dehydrogenase. Alcoholic dehydrogenase. Two enzymes involved here. Now here only one enzyme, that is lactate dehydrogenase. Lactate dehydrogenase enzyme. Lactate dehydrogenase enzyme. Okay. Why did I name my dog Cash here? Because simply like that. Yeah. Next is called as citric acid cycle. Students, what is the other name of citric acid cycle? What is the other name? The other name of citric acid cycle, it is also called as Krebs cycle. Now forget this. I will show you a better one. See here. This is also called as a citric acid cycle. Now I will teach you. I will teach you the entire citric acid cycle in just 1 minute. Ready, all of you? Ready? The small trick is what? The small trick is, officer, can I keep selling sandwiches for money? O in the officer stands for oxaloacetate, C is citrate, I is isocitrate, K is alpha ketoglutarate, S is succinyl coenzyme A, S is succinate, F for fumarate, M for malate. Done. That's all. That's all is the entire citric acid cycle, also called as Krebs cycle. Now, which type of questions can be asked here? Questions. That is very important. Understand, all of you. They can ask you the number of carbon also. Number of carbon. Okay. Number of carbon they can ask you. Okay. Number of carbon. They can ask you number of carbon. Now I will ask you 6, 5, 4, 4, 4, 4, 4, 4, 5, 6. Students, tell me, citrate is how many carbon? Who can tell me citrate is how many carbon? Quickly. Citrate is how many carbon? It's given you in NCERT also. Can you see here? Citric acid is given 6 carbon. But how is the 6 carbon? How is the 6 carbon coming here? 6 carbon. Oxaloacetate is 4 carbon. Acetyl-CoA is 2 carbon. 2 carbon plus 4 carbon is 6 carbon. Isocitrate is also 6 carbon. Now, first this step right here. This step right here. Isocitrate to alpha ketoglutarate. This step right here is the first oxidative decarboxylation. First ODC. Oxidative decarboxylation. First ODC is happening in this step. Now, what is the ODC? Oxide decarboxylation is very simple. In oxide decarboxylation, one NADH is being produced and one CO2 is also released. Can you see? 2 of this will happen in this step. So 6 carbon, 1 carbon is out. So 5 carbon compound. 5 carbon. Now, here second ODC takes place. ODC. Oxide decarboxylation, second one in this step. Alpha ketoglutarate to succinyl coenzyme A. In this step right here, again, 1 CO2 is released, 1 NAD is produced. So 5 carbon to 4 carbon. 4 carbon. Here GDP is produced. What is GDP production? GDP production is here is substrate level phosphorylation. This is substrate level phosphorylation. Substrate level. Oxidation. Substrate level phosphorylation. Only substrate level. Okay, only substrate level. 4 carbon. 4 carbon. 4 carbon. Now here, in this step right here, do you see any CO2 being released? No CO2. So this step right here is a simple oxidative reaction. Okay, simple oxidation, NADH is being produced. Now I'll ask you, in total of how many NADH is being produced here? In one cycle, 1, 2, 3. From 1 acetyl-CoA, from 1 pyruvic acid, 3 NADH, 1 GTP, and 1 FADH, Okay, so from from 1 pyruvic acid, 3 NADH, 1 FADH, and 1 GTP. Okay, all of you should be clear that 1, 2, 3 NADH, 1 FADH, 1 GTP. Now usually for glucose, when we say one glucose has 2 pyruvate, right? 2 pyruvic acid. So this pyruvic acid will produce 3 NADH, 1 FADH, and 1 GTP. Now tell me students, for one glucose, how many NADH total then? How many NADH total? Kasturi, welcome to the channel. Tell me, for one glucose, how many NADH now totally? Tell me, for for 1, 6 NADH. How many FADH? 2 FADH. How many GTP? 2 GTP. For one glucose. For one pyruvate, only 3, 1, 1. Okay, 3, 1, 1. Clear? So for glucose, if they ask you, just add this up. If they ask you per pyruvate, add the single one. Okay. Now students, understand electron transport chain. Very simple. I will tell you briefly. I'll teach you. Okay. Briefly, I'll teach you the entire electron transport chain. Just focus. Ultimate focus for the next 2 minutes. Can we start? Ultimate focus now. Can we start, students? Ultimate focus. Throat clear. Done. Students, quickly smash that like button, all of you. Smash that like button right now. If you're watching recorded, target is 100 likes before the stream ends. We're very close now. We are very close. 97. 3 more likes. The target is over for today. Just 3 more likes. Quickly go. Okay. Now, understand electron transport chain. Okay. Electron transport chain. Ultimate focus now. Maximum effort right now. Okay. Now understand, all of you understand. Electron transport chain is the final step in the respiration. First, glycolysis, Krebs cycle, electron transport chain, ETC or ETS. Electron transport chain is the final step in which all the NADH, all the FADH produced in the Krebs cycle, all the NADH and all the FADH produced in the Krebs cycle is taken up by complex here and converted to ATP. So the main goal, oh, Micah. So jacket removed, mic only gone. Tell me, Micah, now you check, now you tell me. I'll repeat again, don't worry. Suddenly the jacket I removed, no, mic also went off. Didn't realize. I'll repeat again, don't worry. I'll repeat again, understand. All of it's clear now, I guess. It's clear now. Okay. Okay. Understand very well. Understand. Electron transport chain is the final step. Electron transport chain is the final step in the entire respiration. Final step in the entire respiration is electron transport chain. All the ATP and FADH, produced in the glycolysis and Krebs cycle. Glycolysis and Krebs cycle is completely used in the ETC. It is completely used in the ETC. Okay. Now, where does it take place? Electron transport chain happens in the inner membrane of mitochondria. Now, if you look at the electron transport chain, it has 5 different complexes here. 5 different complexes and 2 mobile complexes. Here. Now, I will rotate I will rotate this entire thing, vertical thing, into horizontal. Into horizontal like this. Into horizontal. This entire vertical thing into horizontal like this. Okay. So before we start this, I want you to understand what is a protein complex. Protein complex is a complex which is made up of proteins and electron carriers. There are totally 5 different complexes here. Complex 1, complex 2, complex 3, complex 4, complex 5. Complex 1 is called as NADH dehydrogenase. Complex 2 is called as succinate dehydrogenase. Complex 3, cytochrome BC1 complex. Complex 4, cytochrome oxidase. Complex 5, ATP synthase. I will give you exactly 30 seconds. Remember this. Time starts now. Remember it quickly. I'll give you exactly 30 seconds. Remember it. Once you're done, just say done, sir. Done. 5 different protein complexes. Now, what is a mobile carrier? There are 2 mobile carriers. One is called as ubiquinone, other one is called as cytochrome C. 2 different mobile carriers, 5 different protein complexes. 5 different protein complexes here. NADH dehydrogenase, succinate dehydrogenase, cytochrome BC1 complex, cytochrome oxidase, ATP synthase. ATP synthase. Here, vertical. There. All of you follow me, follow through. Very simple it is to understand. Very simple. Okay, very simple. Now, complex 1, complex 3, complex 2, 4, 5. 1, 3, not 1, 2. Not 1, 2. It is 1, 3, 2, 4 and 5. So 1, 2, 3, 4, 5. 3 will come first, then 2 will come. Then 2 will come. Clear. Okay. Now this is called as inner membrane of mitochondria. This is the entire matrix. This is intermembrane. Intermembrane. Okay, intermembrane space. Now what is happening? First complex is called as NADH dehydrogenase. Yes or no? It is called as NADH dehydrogenase. So in the complex 1, on the complex 1, NADH is coming and setting. NADH is coming and setting here. NADH2 is completely, it is losing electrons and protons. What do you call that? Loss of electrons and protons is called as oxidation. It is losing all the electrons and protons. Okay, so it is getting completely oxidized here. Oxidized. All the protons from here is sent to the FMN, FES, and electrons are sent here. Electrons again sent here, but all the protons are sent to the intermembrane space. Electrons are sent here, protons are sent here. So 2 protons directly from here. 2 protons from this complex is sending 2 more protons from the stroma. So totally 4 protons in the intermembrane space. Okay, that is the complex 1, NADH dehydrogenase. Then there is complex 3. Complex 3. In complex 3, let's get it to later. Let's understand complex 2 first. Complex 2. Complex 2 is called as FADH. Complex 3. It's also called as FADH dehydrogenase. It's also called as succinate dehydrogenase. Complex 2. Okay, succinate dehydrogenase. Here, FADH is coming and setting here. FADH is coming and setting. FADH is sending electrons. Electrons are being sent here. It is also sending protons directly here. It is also sending the protons directly. Protons here. Protons are being sent here. Now you might ask me, sir, here we send protons here, but here why are we sending protons? Why are we not sending protons here? You might ask me, here I send the protons directly to the intermembrane, but here I am not sending. Why? Because this is right here is this protein, this complex is intermembrane. This is inner membrane. So inner membrane, inter. Can you see? It's coming till here. But this one is only present in the inner membrane. Only present in inner membrane. Okay. Now understand. Protons are sent directly here. Electrons are sent again to here. Now what happens here? Understand all of you. Electrons are taken up by a mobile carrier called as UQ, ubiquinone. So ubiquinone gets, once it takes the electrons and also takes the protons, protons, if it's taking electrons and protons, it is getting completely reduced to UQH2. UQH2 against UQ. So it happens in a cyclic manner here. Now once what happens, out of these 2 electrons, 1 electron is sent here. Can you see? 1 electron is sent here. and these 2 protons from the stroma is picked up and all sent here. So 1 electrons, 2 protons from here sent here. Okay, 1 electrons and 2 protons. Similarly, here also UQ, UQH2, UQ. The electrons are being sent here. So from 2 electrons, 1 electron sent here, 2H plus from here is sent here. It is sent here. Okay, 1 electron is sent here. Okay, so in total to complex 3, 1 electron from here, 1 electron from here. 2 electrons are being sent, 2H plus, 2H plus, 4H plus is being sent here. 4 plus being sent here. Done. Now you might ask me, sir, why only 1 electron? Why not both electrons? Because 1 electron is needed to maintain the cycle. Okay. Now from complex 3, the electrons has to be transported to complex 4. Very far. How do you go now? From your house, you're going to a friend's house. You take a cab, take a taxi, bike. Same taxi it is taking. The taxi here is cytochrome C, mobile carrier. So all the electrons are loaded to the cytochrome C. Cytochrome C travels to to complex 4 and releases the electrons. Electrons are sent there. Now once electrons are taken up by complex 4, electrons are transferred. Final electron acceptor. The final electron acceptor is oxygen. The final hero here. Hero in the story. Okay. Oxygen is being taken here. Now oxygen gets converted to water as a byproduct here. Okay. And this entire step, hydrogen, H plus molecules are being sent here. H1 molecules are sent here. Now once enough of H plus is made, here, that is, enough of H plus is made, total how many H plus? 4 plus 4 plus 2, 10 H plus. Out of this 10 H plus, every single time 4 H plus is sent from here to here. Every single time 4 H plus is sent from here to here, 1 ATP is made. 1 ATP is made. 4 H plus from here to here, 1 ATP is made. And that is the entire ETC. Now here I want you to remember even the centers, FMN, FES, cytochrome B, FES, cytochrome C1, FAD, FES, copper centers. This also you should remember. So in examination they can ask you directly the names. This is ATP synthase. They can ask you what is it made up of, how many H plus is being sent. All of these can be a one question. Now students, I know if you're watching this lecture for the very first time, if you're learning electron transport for the first time, it can get confusing. Let me tell you, it can get confusing if you're trying to understand electron transport chain for the first time. I would advise you to re-watch the video one more time. Then if you have a doubt, let me know in the comment section and I will teach you again someday. Okay. Now, next thing is the ATP formation. This should be 4H plus, not 2H plus. 4H plus is being sent, 1 ATP is being formed. Last part of your respiration chapter, that is amphiboic pathway, which explains that any different products are being converted to the final product. And a very simple example I'll give you. If you do not have a food in your house, let's say Sangeeta, Wakanda, all of you, Sony, you do not have a food in your house, what do? You make Maggi or Zomato or Swiggy. Yes or no? You will go for a backup. Similarly, in your body also, there is a favorite food for the body. That is carbohydrates. Our body loves carbohydrates. But if carbohydrate is not available, then does the body starve to death? No. Our body also will look for an alternative. An alternative here is fat or proteins. So fat and proteins can also be used by our body to make energy, and that is called as amphiboic pathway. See here. Carbohydrates is generally carbohydrates is used, but even fats and even proteins can also enter the pathway and produce energy. For example, fats can enter at DHAP and acetyl-CoA also. You can enter at acetyl-CoA. Proteins can enter at pyruvic acid stage. So proteins, because of deamination, proteins can enter here. So basically a backup is there. Body has a backup. Fat and proteins are backup for the body. Okay. Now with that, we come to the last chapter. Of your class 11th one shot revision of diagrams of today's session. That is plant growth regulators. Very simple chapter, very easy. Okay. Now, understand all of you. This is a simple diagram. It is a representation of the apical root apical meristem. Can you see? Root apical meristem, shoot apical meristem. Growing on top, growing on the bottom. That's all. There is also vascular cambium here, present on the sides. Vascular cambium is responsible for the girth. This is a representation of a shoot system. We learned right in morphology. This region is called as meristematic, elongation, maturation. Given here. Okay, same thing. Different zones. Now, there are two types of growth. One is called as arithmetic growth, other is called geometric growth. Now, what is the difference? It's very simple, understand. In the case of arithmetic growth, what happens? One cell is there, it will divide in two cells. When one cell divides in two cells, one cell becomes permanent. One cell is meristematic. Permanent cell will not divide. Meristematic cell again divides in two cells. One cell is again permanent. One cell is meristematic. This will divide again. See, permanent cells cannot divide. Meristematic is dividing again. Two cells. Again, this is permanent. This is meristematic. So, meristematic cells keep on dividing. One becomes permanent. This is called as arithmetic growth. But in the case of geometric growth, one cell divides into two cells. Both are meristematic. To divide two cells, both are meristematic here. Here, one permanent, one meristematic. Very simple to understand. Can you see here? Diagram. If you can notice the diagram, dark blue color is division, permanent division. Here, entire thing is division, meristematic. Okay. Now, this is an example of plasticity. What is plasticity? Plasticity is basically an alternative pathway which occurs in plants' life. That is called as plasticity. Basically, plants are deviating from their goal. Plants are deviating from their life cycle. Okay, that is called as plasticity. It can occur due to the different phase, due to different environmental stress. For example, this is the phase example. Example A, that is larkspur example. Okay, larkspur example. In juvenile stage, the leaf looks something like this, but in adult stage, the leaf will completely change shape itself. So, change in the normal pattern. Other example is buttercup. In buttercup, terrestrial habitat, leaf is very wide because of the, imagine if the water is flowing. If the water is flowing here in aquatic condition, water can easily pass through, less hindrance. So, a same plant in different environment, different types of adaptation. That is plasticity because of environment. Buttercup. This is an example of the curvature test for auxin, a detection of auxin, discovery of auxin. Auxin is located in the tip region here. When we cut the tip, no growth. If I cut the tip and add it on the top, it can grow towards the light. It will grow towards the light. Why? Because auxin is present at the shoot tip and the root tip. Okay. This is apical dominance. If there is apical bud is present, it will keep on growing. If there is no apical bud, lateral growth will happen. No apical bud, lateral growth. Oh, and with that, entire one short lecture is over. With that, entire one lecture. Meristematic cells undergo meiosis. Yes, they divide. Mitosis or meiosis. Okay, usually it is mitosis, not meiosis, because meiosis generally occur in the reproduction cells. Microsporogenesis, megasporogenesis. Okay, our normal mesenchymal tissue is dividing because of mitosis, not meiosis. Mitosis. Okay, so with that, one shot of revision of your entire class 11th botany comes to an end. Now, students, I'm telling you, you have hardly few months left. December, January, February, March. December is almost halfway over. January, February. Students, if you're not joined any kind of crash course, this is your time. And at Vedantu, we have a crash course called as NEET Express. All of you are aware. Second stop is coming. I think it's starting from Monday. So join right now. If you want to join from Monday itself, you can join. Okay. Sir, does the metafloem have bigger sieve tubes? See, metafloem and protofloem are basically different type of phloems. Yes. Okay. So join the crash course right now. Where you'll get unlimited doubt solving. You can get different types of notes, live classes, and also a test series will be given there. So do not wait. Let me know in the comment section how was today's class, and I will see you in the next video. Until then, bye bye. Take care, all of you. Keep studying, keep revising, and kill it. Okay. So bye bye. Take care, all of you. Bye bye. Bye bye, all of you. Let me know in the comment section how was the class. Let me know in the comment.